Hello students. In this module, I will be talking about the process of transcription. Look, listen and learn. So let us recollect the central dogma of protein synthesis. You remember this from the previous modules. You remember this molecule of DNA, which is a double-stranded molecule, correct? Now DNA basically is involved in synthesis of another molecule or another nucleic acid called as RNA or ribonucleic acid, right? It is a single-stranded molecule. Now this process where the information of DNA is transcribed to RNA is called as transcription. Now where do you think transcription will take place in an eukaryotic cell? You very well know that transcription in the eukaryotic cell will take place in that structure which, you guessed it right, is the nucleus, right? It is in the nucleus where DNA molecule will get transcribed into RNA. So how do you define transcription? You can define transcription by saying that transcription is the formation of mRNA on DNA template. Is that okay? Fine. Moving on. In transcription, the genetic information of DNA is copied into RNA. And this process requires a very special enzyme. And the enzyme is called as DNA-dependent RNA polymerase. It basically uses DNA as a template but copies the information into RNA. That's why the name of the enzyme is DNA-dependent RNA polymerase. Now for the process of transcription, we require a transcription unit. A transcription unit is made up of three components, and they are a promoter, next a structural gene, and third a terminator. Now let us see where are these three genes located on the DNA which serve as a transcription unit. Let us understand the transcription unit. For the purpose of same, I'm going to show you a molecule of double-stranded DNA. Now a double-stranded DNA molecule which is involved in transcription has got two strands, yes. Of this, the lower strand which is involved in the RNA synthesis will be called as the template strand, right? Because this is the strand I'm going to use as a template or a mold to build my RNA on. Remember, the template strand is always oriented from 3' to 5' direction. Is that okay? The upper strand, which is not involved in transcription, right? It is not involved in transcription or not involved in RNA synthesis, is called as the non-template strand. Now this non-template strand is also called as coding strand. Is that okay? Yes, and remember, the non-template strand or the coding strand is always oriented from 5' to 3' direction. Now let us see on this DNA where are the three genes located, namely the promoter, the structural gene, and the terminator. Now before that, I want you to assume this point out here. Is that okay? That arrow you see there? Now this arrow is where transcription will begin. So I'm going to call this region as the transcription start site. Now before this transcription start site, there is an area or a region on your DNA which basically provides binding site for RNA polymerase enzyme. Is that okay? And this region is called as the promoter region. So there you go. The first gene or the first area involved in the transcription unit is the promoter. Remember, the promoter is always found on the 5' upstream end of the transcription start site. Upstream meaning before the start site. Is that okay? Yes? Now on the other end, right, you will find another region which basically terminates the process of transcription. Now if it terminates the process, it is also called as the terminator. You guessed it right. Now if you look at the terminator is always seen on the 3' downstream area of your gene. Is that okay? Right? Downstream means every area after the start site. Is that correct? Remember, the promoter, the terminator, now both are given their orientation based on the orientation of the coding strand. Is that okay? Now, in between the promoter and the terminator, you will find the third area or the third gene involved in transcription unit, which is called as the structural gene. So it is the structural gene which is to be transcribed. Is that okay? Controlled by the promoter upstream of it and the terminator downstream to it. Is that okay? Cool. Moving on. Now look at this DNA molecule. Now assume this is an eukaryotic DNA. In eukaryotes, there is only one structural gene, which is controlled by the same promoter and the same terminator. Is that okay? So there's only one structural gene which is getting transcribed at one time. So we say that the structural gene in eukaryotes is monocistronic. Is that okay? Which means only one structural gene. Now assume another DNA strand here. Is that okay? Now let's assume this is the case of prokaryotes. In prokaryotes, there are more than one structural genes that can be transcribed at the same time, involving only that same promoter and the same terminator. Is that okay? Now, because there are more than one structural genes controlled by the same promoter and the same terminator, we can say that the structural genes in prokaryotes is polycistronic. Poly meaning more than one, cistronic meaning structural gene. So remember, in eukaryotes, the structural gene is monocistronic, whereas in prokaryotes, the structural gene is polycistronic. Now this brings us to the end of transcription introduction. In the next module, I'll be talking about transcription, the process, and show you how it really happens. Thank you and take care.